Hi, this is Miss B, and welcome to the vodcast on cell structure and function. In this vodcast, we're really going to look at the interconnectedness between the different organelles that are found in eukaryotic cells, how they work together to complete processes, such as manufacturing and breaking down of organic molecules, also on how they provide structure and support for cells, and how energy is processed in a cell. This would be a great refresher for any level of biology or a great introduction if this is your first time through. So let's take a look at some cells. The first part of this vodcast is going to focus on structures that are involved in manufacturing and breakdown. These include the things that are going to coordinate the manufacturing of various biomolecules, such as proteins, nucleic acids, and the digestion of these things through various enzymes. So first of all, let's remind ourselves that we're looking at eukaryotic cells. And eukaryotic cells are a special kind of cell. They're special in a few different ways. But the key to eukaryotic cells is that we now have membranes. Unlike a prokaryotic cell, a eukaryotic cell is composed of several membranes, membrane-enclosed organelles. So everything from the nucleus, which we traditionally think about, to the endoplasmic reticulum, to the Golgi apparatus, to lysosomes, all of these organelles are essentially containers. They're essentially surrounded by membranes, and what these membranes are doing is they allow for compartmentalization. And compartmentalization is a key evolutionary step because compartmentalization means that you can have different kinds of reactions happening in different places within the cell all at the same time. You can be manufacturing proteins in one area and synthesizing lipids in another. You can be synthesizing lipids in one area and you can be breaking them down in another. So you can now begin to coordinate the activities of a cell so that one cell can be carrying out many specialized functions. Now, over here you see this idealized picture of an animal cell here and a plant cell at the bottom. And notice that it says composite. I think this is important to remember because not all animal cells are created equal. Not all plant cells are created equal. Cells become specialized in multicellular organisms. We call this process specialization. And in specialization, cells gain or lose certain functions. They take on particular functions. Such as your red blood cells. Your red blood cells begin life with organelles and a nucleus and all of those things, but throughout their life they kick out all of their organelles so that they can stuff themselves full of hemoglobin and carry out their specific function of transporting oxygen around the body. Cells in the liver is primarily responsible, well, the liver does a whole host of things. It's actually the largest digestive gland in your body, but one of its important features is that the liver helps break down and detoxify various toxins, poisons, alcohols, things like that that come into our bodies. Because of that, it's going to have a lot of endoplasmic reticulum there. That smooth ER is really important and really essential at detoxifying these things. So you'd find a disproportionate amount of that organelle in a liver cell. A cell that's involved in secreting different proteins, you'd find a lot of ribosomes there because that's the primary function of that cell. So not all cells are created equal, and it's important to understand that when you're looking at a diagram such as these in your textbooks. If we're going to talk about the process of manufacturing and breaking down various biomolecules, the natural place to start is the control center. And the control center of any cell is the nucleus. Okay. The nucleus actually has a few different parts to it. We have the nucleus itself, which is composed of a nuclear envelope, sometimes also called the nuclear membrane, and the nucleolus. Now, the nuclear membrane or the nuclear envelope you can see surrounding the nucleus here. And it's not a solid feature. In fact, it's full of tiny little pores, and you can see these pores right here. This is how molecules get into and out of the nucleus. Now, DNA is too big to fit out through these nuclear pores, which is why we have messenger RNA that's going to actually transmit the information from the DNA out to the factory, out to the machinery in the cell that's carrying on that process of protein synthesis. This nuclear envelope is composed of lipids, phospholipids, like all membranes are, and you can see in this picture here that it's actually contiguous with the endoplasmic reticulum. This is the beginning of what we call the endomembrane system, okay, a system of membranes that transports things throughout a cell. The other important feature of the nucleus is this nucleolus, which you can see on a microscope image of a cell as this dark area here. And the nucleolus has some important responsibilities. The nucleolus is the part of the nucleus that's actually synthesizing ribosomes. Ribosomes, which are going to be our protein factories, and we'll discuss in just a few moments, ribosomes are made of two types of materials. They're composed of protein, but they're also composed of a special kind of RNA called rRNA, which stands for ribosomal RNA. And this is where that ribosomal RNA is produced. Remember that the DNA is all housed within the nucleus, and the nucleus, that membrane, is protecting the DNA. The last thing you want is your DNA to get degraded, to get broken down, to get attacked by various enzymes and things like that in your cell. So housing it in a nucleus really keeps it safe. And that's important because this is the copy of our genetic information that as the cell reproduces is going to get passed on and passed on to each subsequent daughter cell. Now, as we begin our journey through the endomembrane system, a natural place to start is with the ribosomes. Ribosomes are protein factories, and ribosomes consist of two subunits. A large ribosomal and a small ribosomal subunit. And both of these are composed of RNA, again remember that that is specifically rRNA, and proteins. Okay? Ribosomes are the place where protein synthesis happens, and you can find them in two places in the cell. Ribosomes can either be attached, as you see here in this picture, to the rough endoplasmic reticulum, and if a ribosome is attached, it's usually producing a protein that's going to be exported out of the cell or going to be embedded in a membrane. So it'll either be export proteins or membrane-bound proteins produced. Ribosomes can also be free, meaning they're found in the cytoplasm. And free ribosomes produce proteins that are destined to stay in the cytosol. Cytosol is another term that we can use to describe the cytoplasm of the cell. Important prefix there, cyto for you. Whenever you see cyto, think cell. So cytoplasm is talking about the cell. Whenever you see that word cyto, that root means cell. Now, some of these ribosomes, which are attached to the rough endoplasmic reticulum, are going to produce proteins and actually either embed them in the membrane of the endoplasmic reticulum or put them into the lumen, so into this area inside the endoplasmic reticulum here, these cavities, these spaces between there. And the endoplasmic reticulum is really a series of vesicles, so it's containers that are merging in and out of each other. All membranes are composed of essentially the same biochemical molecules, those phospholipids, and so the membranes of the ER can merge with and fuse with the membranes of the Golgi apparatus. The membranes of the Golgi apparatus that bud off of there can fuse with a lysosome, or the membrane of the plasma membrane, the whole cell membrane. So it's important that these membranes have similar chemical structures so that they can merge into and out of one another. Now, we've talked a little bit about that rough ER, and it's rough because it has ribosomes, and this is where protein synthesis is going to happen. But the smooth ER is also an important series of vesicles where different chemical processes are going to happen. And the smooth ER has slightly different functions. Well, first of all, it's smooth because there's no ribosomes, and if there's no ribosomes, what isn't happening here? That's right, no proteins being manufactured here. So what's the point? Well, the smooth ER is involved in the synthesis of lipids. It's also really important in detoxifying poisons. So we find a lot of smooth ER in liver cells. So it's found extensively in liver cells because it's these liver cells, it's the liver itself and the tissue that are responsible for detoxifying so many of these poisons. Both of these are found in the ER, and let's take a look at another picture of them. So here we can see, here's a picture of ER and what's actually happening. I just want to draw your attention to a few points here. So here we have our ribosome, and you can see that that ribosome is synthesizing a protein right here. Recall from previous studies that that protein is not really a fully formed protein yet. It's a polypeptide chain. And recall that a polypeptide chain is a polymer of a sequence of amino acids. So it's composed of amino acids. This is a bound ribosome. It's stuck to the ER here. So we can see that it's synthesizing its polypeptide chain and it's going inside in the lumen, in that inner part of the ER. Once it's there, it starts its process of beginning modification. You know that proteins have multiple levels of structure. So the ribosome is creating the primary structure. Once it's within the ER, it can begin to be folded into those alpha helices, beta-pleated sheets, and take on its final structure. While it's also in the ER, it begins the process of modification. You see certain things added. Often we're adding different aspects to the protein to make it a glycoprotein, glyc meaning sweet, so we're adding sugar change, which might be involved in identification, but then we need to ship it off to its next destination. In order for that to happen, we form a transport vesicle, and a transport vesicle is basically a bubble that's budding off of the membrane of one of these organelles. Because the membranes are self-healing, it can pinch in. Think about it like blowing bubbles in the summer using a bubble wand. And now that transport vesicle, which is really just a membrane with a substance inside, can go and be taken to the next destination, the next place it needs to go to either finish its modification process or be exported out of the cell. Now, we're simplifying this a little bit, right? All of these things aren't just floating willy-nilly in the cell. There's all sorts of highways and byways and things that are actually directing the transport of these different molecules and these different vesicles to where they need to go. This is a great electron scanning image of the ER, and you can see here both rough ER and smooth ER. I think one of the interesting things here is that if you look here, you can tell that the rough ER is rough because each of these black dots that seems to be attached to it, these are all ribosomes. Compare that to the membranes over here of the smooth ER and you don't see those ribosomes attached. That rough ER again is contiguous. It's continuous with the nuclear envelope, and that's really aiding in the synthesis of all these proteins. All right, next stop, the Golgi. The Golgi is really like the UPS of our cell. So the Golgi is primarily responsible for packaging, modifying, and shipping proteins. Okay. It's the UPS. It's going to take them to where they need to go. And so the Golgi is found near the endoplasmic reticulum, near that rough ER. And if you take a look at this diagram down here, you can see that there are two sides. The Golgi is sided. That is that one side receives and one side ships. So a transport vesicle coming off of the ER containing a protein in here is going to merge with the Golgi apparatus on the receiving side. The vesicle merges, dumps its contents into the lumen of the Golgi apparatus, and the Golgi really looks like kind of a series of flattened pancakes. It's going to dump its contents into that Golgi where it further gets modified, proteins might get added, lipid chains might get added to it, and then it will come out of the shipping side of the Golgi apparatus and go on to its final destination, either to be embedded in the plasma membrane. The Golgi apparatus is also responsible. It manufactures lysosomes, which are like the garbage men of a cell. They're involved in the breakdown of old parts that no longer work or molecules coming into the cell. These actually are bud from the Golgi apparatus, or this protein that's been modified might end up embedded in the plasma membrane, in the cell membrane, and be involved in enzymes or signaling and receiving, all those fabulous things that proteins do for our cells. And you can see over here again in this electron micrograph the Golgi apparatus appearing like these series of flattened pancakes here, and you can actually see the different transport vesicles that are budding off and will eventually come in contact with the plasma membrane here. Vacuoles are a group of really diverse organelles. Remember, all these organelles we're talking about are membrane-bound organelles. That's just a fancy way of saying that they're all surrounded by a membrane and they're all compartments in which different kinds of chemical reactions can occur. The beauty, again, of a eukaryotic cell is that you can compartmentalize and have all of these different chemical reactions happening at the same time. So there are a few different kinds of vacuoles. This picture here is of a plant cell. And in plant cells have a large central vacuole. And these are unique to plant cells. In plants, these act as large lysosomes. And in general, all vacuoles act as storage. The other kind of interesting thing about the central vacuole in a plant cell is that think about those beautiful flowers that you got Mom for Mother's Day. Well, you put them in a vase, and if you forgot to water them in a few days, they get kind of droopy. Or that plant, when you get back from vacation, it gets kind of droopy. Part of what gives a plant its structure is water pressure. And that large central vacuole stores a lot of water in it, and that helps give plants its shape and structure. This is called Turgor. Okay, it's the regulation of water pressure. Inside a plant cell. And it really helps to give the plant its shape and its structure. It helps to support it. Now the plant's not going to collapse completely because it does have a cell wall, but if that central vacuole isn't full of water, that plant is not going to be rigid. It's going to wilt. This is just one feature of vacuoles, one purpose in vacuoles. Vacuoles do a variety of other things as well. For example, vacuoles can also serve as just general storage in animal cells. So in animal cells, they're acting as storage. For example, an amoeba, which is a protist, so kingdom Protista, and kind of looks like Attack of the Blob. It is basically a membrane that has all of these what we call pseudopods extending out from it, and that's how it moves around by forcing its cytoplasm into those pseudopods and then pulling its body around. And the way it consumes food is to wrap its membrane around and pull the food inside its body. This is actually a form of digestion. It's phagocytosis. It's bringing the food inside, and it moves it into what we call food vacuoles. So many kinds of protists, including amoeba, paramecium, have food vacuoles as well. A very special kind of vacuole that's demonstrated in paramecium is something called a contractile vacuole, and these are really, really cool. This contractile vacuole is here. Now, paramecium are an aquatic protist, and it lives in fresh water. Just because of principles of simple osmosis and diffusion, water always flows from an area where there's a lot of water to an area where there's less water, from high to a low. And so as a result, and this amoeba living in paramecium living in this aquatic freshwater environment, water is constantly flooding in. No big deal, right? Well, when you're a single cell, lots of water rushing in can become a problem. So all of that excess water gets collected in these contractile vacuoles. And you can see here these different channels that are actually filtering and channeling all the water in. You can see them here. They kind of look like starbursts. Once these contractile vacuoles fill with water, they actually do what they're called. They contract and can pump the water out. So I do have this great video clip that I want to show you featuring contraction of the contractile vacuole. Take a look in this area here, and you should see the contractile vacuole fill with water and then expel it. And this video is courtesy of Pearson. There it goes, contracting and pumping all that excess water out. This is really important for maintaining homeostasis in this aquatic organism. Now we've mentioned lysosomes a few times here, so let's just really take a look at what a lysosome is. Lysosomes are these organelles that are filled with digestive enzymes. They're produced by the Golgi apparatus, and you can see in this picture here the lysosome budding off the Golgi. Now take a second. They're filled with enzymes. What kind of biomolecule are enzymes? That's right, enzymes are proteins. So, because they're proteins, they're manufactured by ribosomes, placed into the rough endoplasmic reticulum, modified, shipped in a transport vesicle to the Golgi apparatus. The Golgi apparatus is then going to continue and finish the final modifications of that, and then these enzymes get packaged in the membrane from the Golgi and are now digestive enzymes ready to go and do their work. They can do things like breaking down old organelles. You can see here in this picture that lysosome has merged with an old and dead mitochondria and is digesting it. They can also be used to break down food that's coming into the cell. See the food particle here coming in via phagocytosis. It's coming in, it's bound in a membrane and bound in a vesicle because it's being pushed in through the plasma membrane. The two membranes merge because they're both composed of phospholipids. They can come together chemically. The contents of that lysosome are dumped into the food vacuole and it can get digested. So why is it important to have these vacuoles? Why is it important to have lysosomes? Well, imagine what would happen if you didn't. These digestive enzymes would be loose in your cell and you'd have no way to regulate what's getting digested, what's getting broken down. So packaging these in lysosomes allows us to control and prevent the cell from getting digested from the inside out. Some cells, when they're going through the process of apoptosis is programmed cell death. And actually a really important part of regulation in your cells. You don't want a damaged cell to go on and reproduce, so apoptosis is what causes that cell to stop growing. Apoptosis is also responsible for your fingers. When you were in utero, you had webbed fingers. There was cells connecting each of your fingers. At one point during development, those cells in between your fingers go through apoptosis and get broken down so that you have separate digits. So apoptosis has a variety of functions. It's really important. When you can't regulate the cell cycle, when you can't destroy a cell that's not functioning properly, and that might have damaged DNA, that's what leads to cancer. So apoptosis is really important. In many cells, apoptosis is triggered when the lysosomes burst open, and then that will trigger the destruction of the cell. So let's just wrap up and review here. We've talked about the manufacturing and breakdown within a cell. This all together is known as our endomembrane system. Endomembrane meaning that all of this is happening within the membrane. Let's just trace a protein production for a minute and make sure that we have this all straight. If our protein begins here as a message, in the nucleus we have our DNA, our double helix. That message gets put onto RNA, which is single-stranded, comes out through the nuclear pores, and is going to attach to a ribosome on the rough ER. The ribosome reads the message, so far we're DNA to mRNA. That mRNA is going to dock with a ribosome. When it docks with the ribosome will read the message and produce a polypeptide. If it's an attached ribosome, the polypeptide gets synthesized and moves into that rough endoplasmic reticulum. Gets modified and moves through in a series of transport vesicles from one layer to the next and will bud off, and you can see here the protein, in a transport vesicle. The transport vesicle then merges with the Golgi apparatus on the receiving side. The contents of that transport vesicle, because the membranes are merging, gets dumped into the Golgi, where it gets labeled, processed, ready to be shipped out. It then can bud off of the shipping side of the Golgi as a transport vesicle, where it may be bound for the plasma membrane and is going to be exported from the cell. It may also become a lysosome if it's full of digestive enzymes, and then go on to fulfill its role in digesting dead or broken down organelles, or even a food vacuole that it comes in contact with. What's really important is to take each of the roles of these different cellular structures and take it to the next level now. Not just the nucleus holds DNA or that ribosomes make proteins, but what's really key here is how do these things work together to fulfill the job of manufacturing and breaking down molecules in a cell.